I hope you can hear online. Speak loud. Okay. Welcome. It's such a pleasure to be here today and be back in this historic room for our Lisa Krivika's Grand Round series. For those of you who don't know me, my name is Sabrina Paganoni, and I am an ALS researcher in the NGH ALS group. And I would like to welcome everyone who's here in person, as well as those of you who are here online. So who was Lisa Krivikas? Well, I want to start by introducing why this Grand Round series is so special to me and to many in the audience I see here. Lisa Krivikas was an ALS researcher, an associate professor in the Department of Physical Medicine and Rehabilitation at Spalding Hospital and NGH. And I met her when I was a resident in 2008. She was a great teacher, and all residents were drawn to her, including myself. So I asked her to mentor me, and little did I know that that encounter would change my career. So a few months after I met her, she gave a very powerful Grand Rounds lecture where she announced that her ALS research had become personal. She herself had developed ALS. In fact, she died 1 year later before I finished my residency. But before that, she continued to mentor me, and she encouraged me to continue her work as a physiatrist in ALS. And so she introduced me to Merit Sukovic, who's here, the chair of our department, and she encouraged me to work with her. In fact, she encouraged me to do a research elective as a resident in the NGH group. So at the time, there were a few residents there. There was also a fellow named Dr. James Berry, who was there. And he was starting his career in ALS research and later became a great mentor, colleague, and friend. And so, as we continue to honor the legacy of Dr. Krivikas, I want to say a few words about what she wanted to do. She wanted to do something meaningful to change the course of ALS. And while we don't have a cure yet, we certainly have had a lot of progress, especially in the last year, with 2 new approved drugs for ALS. There's also 1 drug that's up for approval in 2023 for genetic forms of ALS, which is so important. And so a person who has been at the center of all of this, in addition to Dr. Sukovic, is Dr. Berry, who is our speaker today, who has made seminal contributions to the field of ALS, has been involved in many programs that are connected in different ways to drug approvals for ALS, and has really been at the forefront of ALS research. So let me read a couple of things about Dr. Berry. He's the chief of the division of ALS and motor neuron diseases. I'm sorry, I have to read it because there's so many titles and so many accomplishments. I just need to remember the main ones. He's also the Winthrop Family Scholar in ALS Sciences. Is the Averill Healey Endowed Chair in ALS. Is the director of the Neurological Clinical Research Institute. And among his many accomplishments, Dr. Berry is an international leader in ALS biomarker development and clinical trial methodology. So there are many things that I admire about James, and you can read the rest of his bio in the program. But the one thing that I would like to highlight today, and that I really admire, is that he's a visionary leader. He's the one who sees where things are going before everyone else does. And that's reflected in the title of his talk today, which is really a forward-thinking talk. Today, we'll be talking about digitizing ALS, using digital data to quantify behavior and function in ALS and hasten drug development. Before we give the podium to Dr. Berry, I would like to present him with a plaque to honor again his Dr. Krivika's legacy and these lectures.
Thank you. Thank you all for being here. Oh, a little technical issue. All right. Thank you all for being here. Sabrina, thank you for that very kind introduction. It is really an honor to give the Lisa Krivikas lecture and to hopefully honor her legacy of mentoring, of teaching, of research, and of clinical care. And really at the Healey Center and the NCRI, we are doing so much work. There's so much going on that just to select the topic was somewhat difficult. But I wanted to talk about something that really spins out of a fairly simple question, which I remember I really started toying with primarily. I remember walking away from one of our Ice Bucket Challenge events And this question was sort of on my mind, burning on my mind. And I ran into an old colleague who was in psychiatry, but we had been residents together. And we started talking about what if we could use digital devices to quantify people's function, hasten drug discovery, and reduce trial burden for people with ALS and other neurological diseases. And that was sort of the genesis of a very simple question that spawns really all the work that I'll be talking about today. It's a little bit out there as a question, but then I sort of thought, well, you know what, in cardiology, we've been doing this with Holter monitors and King of Hearts monitors and all the monitors that have spun out of that for a very long time. And it's really not that big a leap to say we'd like to do this for bulbar function and strength and respiratory function in ALS. And so it went from sort of a big kind of out there question to actually just an iterative question, but one that I think is really important. These are my disclosures. I do try to be involved in many of the projects that are going on in ALS. Because this is sort of couched in ALS, I'll say a few words about the disease that I think will be important to frame the rest of the talk. It's caused primarily, though not exclusively, by a loss of motor neurons in the brain and the spinal cord. Certainly, there are other neurons that are affected, which is why 5% of people develop FTD, and many of the supporting cells like microglia are affected as well. The classic clinical tagline is a progressive painless weakness, and I'll just pause to say that at the outset of the disease, this is a good description, but people with ALS often bristle at this because pain does become a part of the disease for a variety of reasons: immobility, spasticity, injury. And so we want to be cautious about how we think about that. It can start in any region. The bulbar region, cervical, and lumbar region are about equally represented for the onset of the disease. And then 1 to 3% of the time it starts as a respiratory disease. And the average survival after symptom onset is 3 to 5 years. However, there's a wide variability, with 10% of people living longer than 10 years and some people succumbing much faster to the disease. And that variability is both troublesome as we do research, but also I think something that we can learn from. There's a diagnostic delay, that is about 12 months pass from the time that symptoms start until people get a diagnosis on average. And this is actually remarkably similar around the globe and over time. And it's something that we'd like to make progress on. And I'll touch on that in a sort of in a way later in the talk. As Sabrina mentioned, we have 3 FDA-approved disease-modifying medications: riluzole, edaravone, and sodium phenylbutyrate terso. And we have myriad symptomatic therapies as well. But in addition, we have a very full pipeline for clinical trials. In fact, so full that doing traditional design clinical trials is really not going to serve us. We need to test things faster, which is leading to innovative trial designs. And I'll touch on that as well. But just to give you a sense of how disease-modifying therapy discovery is hastening over time, up until 1995, there were no approved disease-modifying therapies. We had riluzole approved in 1995. It's an anti-excitotoxic medication. In a very general sense, leads to about 10% slowing of the disease. And then we had a long pause in terms of approvals until edaravone, an antioxidant, at least in theory, showed 30% slowing in one of 2 trials. And gained approval by the FDA in 2017. Now, this is an IV formulation taken 10 days out of the month. And it was no small breakthrough then to have oral edaravone approved in 2022. Also in 2022, in a trial led by Sabrina Paganoni, sodium phenylbutyrate and terso in combination was shown to have a 25% slowing of function, 25% slowing of function decline, as well as a survival benefit. And there's a global phase 3 trial ongoing, not in the US anymore because of the approval. But you see how these dots begin to sort of pile on top of one another. And as Sabrina mentioned in her introduction, tofersen, which is an intrathecal antisense oligonucleotide for SOD1 ALS, is in front of the FDA now. And it has been shown to reduce SOD1, to reduce neurofilament light, and to slow disease, though it missed its primary endpoint in the randomized controlled trial. Secondary endpoints were positive, and there was a slowing of disease in the open label extension. So we're hopeful as a community about that. And not only that, but there are many, many ongoing clinical trials, more than ever before. And so, we are looking forward to this continued pace of discovery. It's in that setting that Merit started the Healey ALS Platform trial, and she is the PI, Sabrina is the co-PI of this. And really our whole organization is contributing to this. It's very complex, but it's also really moving us towards answers faster. And so we've completed 4 regimens. We've released results on 3 of those regimens. A 5th is enrolling now, and we have 6th and 7th lined up and others for design phase after that. There are efficiencies to this in terms of how we do startup, how we interact with sites, how we enroll participants, and we reduce the placebo burden. But this trial, like other trials, really rely on traditional ALS outcome measures. The revised ALS Functional Rating Scale is a questionnaire, not unlike the UPDRS and Parkinson's disease. It has 12 items. It's delivered by a trained examiner in clinic or over the phone. And then the participants' answers are interpreted and scored. It has a respiratory subdomain, a bulbar subdomain, a gross motor subdomain, and a fine motor subdomain. And this is really the workhorse of our clinical trials. It really takes on the importance of the modified Rankin scale for stroke. It is sort of a primary endpoint across the board, basically. We do have also secondary endpoints like vital capacity, which measures strength of the muscles of respiration, quantitative motor strength testing, and survival. Survival is obviously a very key endpoint, but we try to do trials that are shorter. So over 6 months, it's very difficult to have statistical power to look at survival as the primary endpoint. This sort of leaves room for improvement. I want to just take a little look at the ALS Functional Rating Scale. These are the subdomains and the questions within it. So in the bulbar region, we're looking at quantification of speech, of salivation, of swallowing. Fine motor is handwriting, cutting food, dressing, and hygiene. Gross motor is turning in bed, walking, and climbing stairs. And in the respiratory section, there are questions about dyspnea, orthopnea, and use of BiPAP. Now, this was really designed from a clinical perspective. These are really some of the key things that we ask about in clinic, and it captures many of the parts of the disease really well. It's quite interpretable when we look at kind of the domains that we're covering. However, it wasn't really designed as a psychometric tool in the way that we think about it now. So rash analysis shows that the items overlap, that the item weighting is not uniform, and that the scale is not unidimensional. These are all things that we would look for in a sort of a well-designed psychometrically sound tool these days. Having said that, it actually performs remarkably well for a scale that looks this sort of out of bounds for a rash analysis. So I will not be saying throughout this talk at all that we should be throwing the ALS-FRSR aside, but rather we should find new ways to look at it and ways to supplement it. One other thing about the ALS-FRSR is that its progression is treated as linear, but actually it's more complex. So this is a figure that comes from a paper from Divya Ramamurthy, a doctoral student at MIT. And it shows not only is it not a linear decline, but actually we can cluster people by different shapes of decline. One thing I would point you to here, though, is that this tells us something about the disease and the interaction of the scale and the disease, but the time course here is over 3 years. And I just told you that we try to do 6-month studies. And so if we pick out almost any 6-month epoch in this, it actually is declining in a fairly linear way. So I don't think we want to be too hard on our scales that exist now, but there are opportunities to analyze the data we have differently and analyze different data. So there are many additional scales. Some are covering similar topics to ALS-FRSR, and some are covering important unmeasured impacts of ALS. And I think we have an opportunity to look at those. And we also live in this very data-rich world, and we should take advantage of that. And I think there are ways that we can do that we've really left on the table. Some that I talk about today are fairly well mature. Some are more exploratory. But digital biomarkers present this opportunity to quantify function, to gather big data. They can be simple to do, simple to include in trials, and they can facilitate those trials if we do them right. They can also inform care. I have to say that my perspective today, especially for today's conversation, but really in what I do, I'm framing things through the eyes of a clinical trialist. So digital outcome measures are at sort of a certain standing in ALS. That is, researchers, people with ALS, and you'll see this abbreviated as PALS throughout the talk, and regulators would like to move toward digital outcome measures and trials. The goal being, I think from everybody's perspective, faster trials, less burden, and something that's more reflective of real life impact. But we also don't want to run into this naively. In order to be worth adopting, these outcome measures have to do something better than we're doing currently. It may be that they get us to an answer more quickly, that they are more reflective of real life. It might be that they're easier to administer, or they give us a broader view of things, but they have to do something better than what we're doing in order for us to adopt them. If we do find that, then there are a few steps we need to go through to adopt these digital outcomes. We have to gather data to show that they're useful, and we have to feel comfortable about usability and compliance. And that's no small bar in some ways, and I'll show you some data about that. We have to understand their change over time. So there's, I think, an inclination many times to say, oh, look, we can separate ALS from healthy volunteers. Isn't that a great scale? But we really need to look over time in order to adopt it, though cross-sectional analysis is a good first step. And finally, there are regulatory considerations. And I think there are groups like a nonprofit, Everything ALS, as well as Critical Path Institute. And FDA itself has some divisions that are working with investigators to try to incorporate digital biomarkers into trials. To give you a sense of one of the ways that these promise to be good additions to our traditional outcome measures really provide sparse data. So let's say we do a 6-month trial and see people every month during that time. We'd have 6 data points on our primary outcome measure. So if somebody misses one visit, we're now missing almost 20% of our data. Using what we call active digital monitoring, that is asking people to fill out a survey or do a task or make a speech recording, we could easily ask people to do things every week, and we get a much more robust data set that's more resilient to data that's missing. And when we move to what we call passive data monitoring, we get really large data sets. And this is looking at data that comes from sensors while people live their everyday life. So it could be a wearable or it could be data coming from sensors on a phone. And this is truly a remarkable change in the size of data set that we get. That's a benefit of passive data monitoring. But there is also something else that we have to be careful about with passive data monitoring, which is that we have voluminous data, it's highly variable, and it requires a lot of processing. And the algorithms that we use to process that become the main part of what we need to research, because we need to have a huge amount of trust in our algorithm. So this is not raw GPS data, but it's unfiltered GPS data. And when we put that through an algorithm, we can see that here, the distance that people are traveling over time declines as the disease progresses. But we have to have a lot of confidence that that algorithm that we're using is doing its job correctly. And that's true of sort of all of passive data monitoring. So with all of this in mind, we set out to do a study, which turned into a series of studies that we called symptom monitoring for ALS in real time. And this uses something called the BIWI smartphone app and data portal, which was designed at the School of Public Health by J.P. O'Nella. And BIWI is an end-to-end encrypted, very secure platform for collecting both passive and active data on a cell phone. That data is collected and sent to the cloud where it's stored as raw data. And there is also a cloud processing forum that holds standard processing algorithms so that they can be applied to the raw data, applied to other data sets, and new processing algorithms can be added to this so that you can reprocess raw data. This allows us, which is very important in academia, to analyze data and then to replicate those analyses with exactly the same parameters. But it also allows us to reanalyze data differently. And that's really important when we're generating these huge data sets, because we get better and better ideas, and we've seen this happening over the years, about how to analyze this data. We did this on personal cell phones, so people put it onto their own cell phone. And we did that because personal cell phones present an opportunity. There is truly a condition called nomophobia, which is a fear of not having your cell phone with you. It's actually being considered for the DSM, but in a very mild form, lots of us have it. And so as a result, people have their cell phones with them. So 90% of adults have their cell phone within arm's reach throughout the day. But not only that, smartphones are loaded with sensors, accelerometers, gyroscopes, GPS. And also we can ask people to do active data tasks on them. So fine finger movement tasks, cognitive tasks, speech recordings. Phones have remarkably good microphones built into them, and patient-reported outcome measures can all be obtained. So we designed a study where we got ground truth data with sort of in-person data. Because of the pandemic, a lot of this is done by telephone, but the ALS FRSR delivered by an examiner, done at 0, 6, 12, and 24 weeks throughout the study. And then we got active app data. So we had baseline surveys, including one I'll tell you about in a moment called the Communicative Participation Item Bank, as well as the SelfEntry ALS FRSR. And we got weekly recordings of both speech, people reading a passage, a standard passage, and a voluntary cough or volitional cough recording. And then we also got all of the passive data from the phone. And this is, by the way, can be done on both Android and iPhone, which is unusual for programs and a real benefit. One of the criticisms that we got when we set out to do this was, look, something like GPS is a very blunt tool. Will it show you anything? And the world sort of provided us an opportunity to look at this, because around the time of the COVID pandemic onset, we had about 8 people with ALS using this program on their cell phones. And we just took a quick look. We said, look, the world has changed dramatically overnight. And if we can't pick up that relatively large change in behavior, then we're probably in trouble with GPS. But if we can, it's a good proof of concept. So it turns out that our group of people with ALS was spending about 19 hours a day at home prior to the onset of the pandemic. We used the state of emergency in the US as kind of a delineating line. After that, in the just month or two after that, those same people were spending almost 24 hours a day at home. And so, big change in behavior, we certainly can pick that up. It leaves the question, will this be useful for more subtle change, like a change in behavior from ALS? But I think it is a proof of concept. Now, there were some other large databases of just general population changes, and they saw a very similar absolute change in how much time people spent at home, but it went from 10 hours to 14 hours a day in that sort of 1 month period. And so even though people were saying, oh my gosh, I'm never leaving my house, I'm never leaving my house, the reality is they were still spending less time at home than people with ALS were spending prior to the pandemic. One of the benefits of BIWI and smartphones is that we can obtain audio on that platform. And I've been working for many years with Jordan Green and Kate Conahan at the Institute of Health Professions at MGH, and they do very sophisticated motion capture and studio recordings of audio to get at speech diagnostics and develop quantitative motor speech parameters that may help us follow disease. And in ALS, it turns out that pause time in a read passage, so we have a standard passage called the bamboo passage, we have people read it, and the amount of time that they spend pausing is actually an accurate early diagnostic marker of bulbar onset of disease. So if we do this in people who have ALS, but do not complain of any bulbar symptoms, and we don't detect any bulbar symptoms, we can actually still separate them from healthy volunteers based on how much time they're pausing. There are other parameters we can get out of this, like speaking rate and articulation rate, which are also very useful parameters. In this cohort, at least, pause time was the most sensitive. We applied this in a trial some years ago of dextromethorphan and quinidine. Now, this is approved for pseudobulbar affect, but there was some thought that it may have an effect on speech as well. And applying these same parameters, we see this was a crossover trial. So there was an active period and a placebo period for everybody. They were randomized in what order they went in. Here, I've just separated them into active and placebo. But the percent pause time increased during treatment with placebo, but decreased during treatment with dextromethorphan and quinidine, suggesting that there is an effect of dextromethorphan and quinidine. In a more detailed analysis, it looked like there were some people who responded to this and some people who didn't. And I would just say clinically, that's sort of what it seems like in clinic, that some people don't have much response, but a few people do. That improvement in percent pause time was really driven by longer speech segments and shorter pauses between those. So we can really even drill down into sort of why are we seeing this change when we have these really large quantitative data sets. Now, that is all in a highly controlled clinical setting, but we ported that over to use it on a smartphone and make recordings. And there were a lot of questions about this. Is background noise going to be a problem? What about the microphone itself? Is it good enough? What about the fact that people can have this microphone close to them or far away from them? And that does limit some of the kinds of analyses that we can do. But as far as sort of pause time or speaking rate, it actually has been very robust to this. And Jordan and Kate Conahan have been really leaders in showing how we can clean up the data and do these analyses. And so we had people read a standard passage, and we looked at their average pause duration, the total time they're not talking during that passage divided by the number of pauses. And that average pause duration increased in all but one of our participants in this pilot study, which was really encouraging and has been replicated in a number of analyses. And this really has led us to put this as an outcome measure into the platform trial and other trials, which is really exciting. Now, Kate Conahan had the idea, and I think it's a very, very important one, that one might look at this data and say, okay, so people are pausing more or they're speaking more slowly, but does that matter? And that, I think, is going to be a response of the FDA if we went to them with the data that we have. So we incorporated this communicative participation item bank into our studies. And this is a 10-item questionnaire that asks, how do people use verbal communication with people they know, with people they don't know, for simple topics and for more complex topics? And what we found is that when people are speaking at a normal rate, there's sort of an agreed upon threshold, basically agreed upon threshold. Is anything really agreed upon in medicine? But basically agreed upon threshold of 150 words per minute as a normal speaking rate. If people are speaking at a normal rate, they rated their communicative participation as normal. But if they're speaking at a slowed speaking rate, most of those people rated their communicative participation as abnormal. And this, I think, imbues that quantitative data with a real clinical meaning and is remarkably important. So Kate has much more data with this now and is working on a paper, which I think will be really important. We were also making cough recordings and looking at a cough audiogram. This is a volitional cough, so we asked people to cough. And we thought, cough involves respiratory system and the bulbar system, and this may give us an insight into both of those systems. When we apply a machine learning model, it actually turns out that we do have a moderate accuracy for detecting bulbar symptoms, but really not a very good accuracy at all for respiratory symptoms, which was a surprise to us, but I think is an interesting finding. We want to validate this work with clinical perceptual rating. So basically having people listen to the cough and rate that cough. And there are rating systems for cough that we can do that with, and we have the recording. So I think that's a good next step. We'd also like to improve the model. And Bridget Perry is in Jordan Green's lab and is really working on this. But it raises the question, and we're only at the early part of this, and we have longitudinal data we have to look at. Can volitional cough serve as a marker of bulbar impairment? And I think it's a really interesting idea. We can also use the same platform and other platforms to deliver traditional scales, but to be used as patient-reported outcome measures. And so I told you that the ALS-FRSR has to be delivered by an examiner, but the SelfEntry ALS-FRSR, just having people answer these questions on their phone, it turns out correlates very well with the examiner-delivered or traditional ALS-FRSR. We do have to note that the SelfEntry scale is higher. So people rate themselves about 2.4 points higher, 2.5 points higher than the examiner rates them. But the correlation is incredibly high. Now, this is a pilot study. The next question that you have to ask is what happens over time? So we looked at the slope of the in-clinic ALS-FRSR, the traditional one, and the smartphone SelfEntry ALS-FRSR. And it turns out that the mean rate of decline is actually remarkably similar between the two, certainly not statistically different. We get a lot more data for SelfEntry because we can ask more frequently. And the standard deviation as a result is smaller for the SelfEntry ALS-FRSR. So I think that's incredibly encouraging as well. We wanted to replicate these results in a less savvy population. We were concerned that perhaps people who would enroll in a study like a digital biomarker study might be more familiar with research and know the scales. So we went to our clinic and we used the patient-reported outcome measures that we do in clinic before people come into clinic, sort of as a standard part of our clinic. They're doing these outcome measures on an iPad, and then we had people come into clinic and we had the examiner deliver the ALS-FRSR. And when in this completely unselected population, when we compare those two results, we see that there's a very high correlation. There's a little more spread to it. You see a few outliers, and I think that's something we have to accept with the SelfEntry scale, that there will be a few outliers. But the correlation is very high, and we see almost exactly the same offset. So people continue to rate themselves a little bit higher than the examiner rates themselves, but in a reliable way. We don't have as much longitudinal data from this, and I'm not showing it here, but it showed decline as well. Interestingly, when we look at this as a SelfEntry scale, we can get more information as well. So it turns out that lower ALS-FRSR scores correlate with a longer ALS-FRSR completion time. Now, I don't think that this is probably a robust enough variable that it could be an outcome measure in clinical trials, the time to complete the survey, but it could be potentially a covariate. It also, I think, raises a really important question, which is, or point, which is that as the disease progresses, doing any of these outcome measures gets harder for people. And that is, I think, important from a humanistic perspective. It's important for how we design trials. And it's also really important for digital outcome measures when we think about compliance. This is something that is not written about in digital medicine as much as it should be, but compliance is a real issue that we need to deal with in these digital outcome measures. So these are from 3 different studies using the BIWI platform. Study 1 was a 12-week study, SMART ALS study. We then went to a longer observational study with about 100 people that we call study 2 here. And study 3 was a trial of inosine where we implemented this, a randomized controlled trial. So we saw very good compliance in study 1. GPS, which is the only passive measure I'm showing, compliance was a little bit higher than the active data collection for audio and surveys. In study 2, where we had more people, maybe a little bit less kind of the vanguard, the early adopters, we saw that there was a steady drop-off in compliance, which is actually better than what's published in the literature if you look closely, but still, I think, left us really saying, we need to do more. I will also make the point that we did absolutely nothing to remind people about this. We put the app on their phone at a visit, and then they just did whatever they were going to do with it. It had reminders that came to them. But we thought, I think we can do better than this, and I'll show you some data about that. I think encouragingly, in the trial of inosine, we actually see very high compliance. Now, again, that's a shorter study, and it may be that when people have a shorter target, they do better. Interestingly, we also continue data collection for the safety period of 28 days after the end of the treatment, and you see the compliance begin to drop off. So it's a good thing, I think, that we have some evidence that people will comply better in a trial setting than in an observational setting. And that's true, I think, for other biomarkers as well. But we learned a lot from doing that. And I think it's really important for the field that we're honest about compliance. So the last SMART study we did, we added a wearable component to this. So the remainder of the study is basically the same. And we added either for participants, either an ankle worn wearable called the stepwatch or an actigraph wrist worn wearable. And I'm going to show the actigraph data here, although they both did a very good job of counting steps. But what we found is that step count and activity count, which is sort of like a somewhat more sophisticated step count, both decline over time and correlate with the ALS-FRSR. So you can see all the way on the left that the number of steps declines. It's a broad scale, so we maybe should look at log steps so that it can spread out a little bit. But those steps decline over time. We have to correct for non-wear time. So if people are less compliant over time, it'll look like their steps are declining, but we do have ways to analyze that as well. It turns out that was actually easier on the actigraph than the Motus stepwatch, and that's part of the reason I'm showing this data. If we compare it to the ALS-FRSR, we see that as the ALS-FRSR is lower, there are fewer steps as well. And that's true of total activity counts as well, which is, again, just a sort of a proxy for step count. Now, we also got actigraphy data from the smartphone itself. This is a very different algorithm or pathway. It seems like they're counting the same thing. The truth is we're not actually counting continuously from a cell phone, so there's a lot more interpolation that has to happen, but we saw a very similar thing. First of all, using GPS, looking at home time, we see that people with lower ALS-FRSR scores spend more time at home. We see that the distance that people travel is shorter when people have lower ALS-FRSR scores. And then if we look at the log of activity index, which is an accelerometry-based parameter, we see that people with lower scores have less activity. So with all of this in mind, we went into another study. This was with a partner, Biogen. We called it the Digital ALS Study. The catchy name came from the coordinator, SMART. The Digital ALS Study, that came from me, brute force. But we provisioned phones to people. We had a sort of highly curated user experience. It was well-resourced, so we had coordinators many of whom are here actually, who could call participants and keep them involved in the study and give them reminders. And we were really focused on active data collection for this study primarily. So the basic outline of it was a 25-week study. It was observational. There were 3 visits that were phone visits at baseline, halfway through, and at the end. And people were doing these digital assessments every week throughout. And we had 19 people with ALS and 23 healthy volunteers. So the kind of things that we were asking people to do were patient-reported outcome measures, cognitive assessments, which I think we have some more work to do on, to be honest, and I'm not going to present those results. But I think mainly what we learned is we need to revamp our cognitive assessments. Speech recordings, fine motor or dexterity measures, and walking and balance tasks. So the first thing to say is that compliance in this study was much, much better. And I think that is testament to the fact that people want to do this right, but they need reminders throughout it. And this was a lot of work for our coordinators. They had to track in real time what compliance was like and make some calls to remind people. But I think this is a good sign for the future that we know how to do this. The other thing to say is that the traditional and self-mentored ALS-FRSR again correlate very highly. Again, people rated themselves a little bit higher, not quite as much higher here, about 1.5 points, but still very high correlation. And then we looked over time and we saw a very similar story to what I showed you before, which is that we have much more data, but the trajectories look very similar. And what I show on the right-hand side is that the point estimate, here's my pointer, the point estimate of the rate of decline of ALS-FRSR is actually really consistent across traditional every 3 months and the digital measures. Now, what we did is artificially subtract some of those measures and say, okay, we gave it weekly, but what if we only look at every other? What if we only look at once a month? Or what if we look at every 3 months? And what we see is that even though the point estimate is the same, the confidence interval goes down as we ask more frequently, so that we have a lower confidence interval, smaller confidence interval, for weekly examinations with ALS-FRSR than we do for the 3-month, even though it's traditional. That means more statistical power in a trial, and we could leverage that. The other thing that I would say in looking at this is that there is a little bit of the law of diminishing returns if we look at every 2 weeks compared to every 1 week. So yes, we do get a smaller confidence interval, but it's not very much smaller. So it might be reasonable to ask people to fill this out every 2 weeks and get additional power. Now, behind loss of strength, fatigue is the second most common complaint from people with ALS about their symptoms. And the Neurological Fatigue Index for Motor Neuron Disorders was developed by Carolyn Young in the UK. We applied that in this study, and what we found, which is consistent with what people tell us, is that people with ALS compared to healthy volunteers had much higher levels of fatigue. And you can see that in the summary subscale, weakness subscale, and the energy subscale. And I think, you also see that some people who are healthy volunteers have fatigue. Some of these people are caregivers for people with ALS, and that might explain it, but some people are just have fatigue. That's not surprising. I think it'd be surprising if we didn't see some of that overlap. But I think it highlights how important this is. The next question we ask ourselves, again, is what happens longitudinally? And it turns out that actually there is a slope to this. The fatigue increased over time. And so this may be a measure that we want to monitor for drugs to see if we can have an impact on fatigue. And we're doing some, I'm not going to present them today, but some studies to try to uncover what is this sense of fatigue versus muscle weakness versus the fatigue of muscles. And we have a study using digital assessments, sort of bringing people into the office to try to get at the connection between those or dissociation between those. Now, we also thought that mood might affect this, fatigue scale. So we looked at that, and there may be a small effect, but certainly not statistically significant. We also looked at morning versus midday versus evening. When did people take the scale? Suspecting that if they took it in the evening, they would report more fatigue. It actually turns out that they report more fatigue midday, though whether that, and in fact, evening versus midday is statistically significant, but barely. And I think, having more people would be interesting. But I think, that's actually reassuring to say it's not changing dramatically based on when they take it or what their mood is. We looked again at speaking rate, and this correlates with the ALS-FRSR bulbar subscale. And one thing I wanted to point out here is the y-axis is the speaking rate in words per minute. The color is the max pause time. And you can see that those two things are really highly correlated. So that lower on the axis, that is slower speaking, correlates with a higher pause rate. But also that we do see this decline in speaking rate as people report more dysfunction on the bulbar subscale. A finger tapping composite score, that is total taps and the time between taps, correlates with the ALS-FRSR fine motor subscale. We want to look at this over time. We haven't yet done that. But I think this is really tantalizing evidence that we may have measures to quantify fine motor movements as well. And then finally, we looked at gait speed. And overall, gait speed does correlate with ALS-FRSR gross motor subdomain. You'll see how the healthy volunteers kind of are over at the 12. There's a fairly big spread there. Interestingly, we see a fairly stable sort of from 7, 8, 9, 10, 11, 12 on that subscale. We see a fairly stable gait speed, and then it begins to decline below that. But the pink dots are people using walking aids. And our algorithms really are not set up for people using walking aids. They're not validated in that population. So I think we have some more work to do on this as sort of an active task. And that's okay. That's one of the things you learn in these early studies. Now, we've been working with a small company called NQ Medical on a different way of looking at fine finger movement. So NQ Medical has a way of exploring and quantifying fine finger movements. And that is that they store and analyze the characteristics of how people type, but not the content of what they type, from a smartphone virtual keyboard. So just like you can add a keyboard for a different language or a swipe keyboard on your cell phone, this is a research keyboard that you add and you can select using that. And so participants put this keyboard onto their cell phone and they type on it, and it records things like how long do people press on the letter? How long does it take them to go to the next letter? How close to the center of the letter do they type? How often do they use the backspace? And then they develop algorithms, and these algorithms already can distinguish Parkinson's from controls and on states from off states in Parkinson's. And so we wanted to see if we could do a similar thing in ALS. And indeed, we can see that using what we call the NQ index, which is sort of the number that comes out of this algorithm, we can differentiate ALS from healthy controls to quite a high degree. Now, some people with ALS don't have much fine finger abnormality, so it doesn't completely separate, but it's at least as good as any of our biofluid biomarkers. High sensitivity specificity. We did an alternating finger tap in this study as well, and that did not have nearly the sensitivity. In fact, not good sensitivity at all for separating healthy controls and people with ALS. Then we focused on the ALS group, and we asked, does the NQ index correlate with ALS-FRSR, as we've done before? There is a correlation. This is the total ALS-FRSR score. We wouldn't expect 100% correlation here, but I think on the face of it, it is encouraging that there is a correlation. And then we again look longitudinally. So what we did here was took our group of people, and this is an interim analysis. We actually have our final data set now. So the coordinators did their final visit with the final participant just last week, I think. But in this interim analysis, we took the group of people who had longitudinal data, we divided them based on the rate of decline of their ALS-FRS-R, so that in green you see the slow progressors and in red the fast progressors. And then we just calculated the slope for those two groups. And because that's how we made our groups, clearly there's a separation in those two slopes. What we wanted to do then is apply the NQI and say, does the NQI have a different slope of decline amongst slow progressors and fast progressors for the total ALS-FRS-R? And indeed, we do see a difference. So what one might look at this and say, well, look, the separation is more on the ALS-FRS-R, that's a better scale. But the reality is we made our groups based on the ALS-FRS-R, so there has to be more separation there. That's how this works. But this does show if we have a slow group and a fast group, we can detect that using the NQI. And so if we had a trial that slowed progression, we would end up with a slow group and a fast group, and it would look something like this. And I think that's very encouraging. So I'm really looking forward to the final analysis from this. I want to turn my attention briefly now from developing markers that really fit nicely, I think, into clinical trials if we get to get a little more data on them, to a little more, maybe we'll say out there sort of an idea, which is, can we use internet search data to identify people with ALS? And I understand that this is a sort of a unusual question. It really comes from the fact that there is this long diagnostic delay that has been stubborn. We have had a hard time reducing that diagnostic delay. We've embarked on various projects to look at charts very closely or use natural language processing for medical records that have not really yielded good results. And part of the reason for that is we're looking at the same data that we have in front of us anyway. So what I thought is, maybe we could look at data that is not a part of our health system. Maybe there's a data trail that would lead us to understanding somebody was developing ALS. And I got laughed out of a lot of rooms when I sort of said that, probably appropriately so, but eventually Ernest Frankel, who's at MIT, said, actually I have a friend who's working on this at Microsoft Research, this kind of thing, and he would be a good collaborator. And this comes from Elad Yam Tov. The idea is that, look, we're putting trillions of data points into the internet at any given time. And that is a completely new source of data, and we can plumb that for medical purposes. And he actually had already done work saying, search engines can track real life events. So he looked at pregnancy. And early in pregnancy, people are searching for things like late period faint line pregnancy tests. Mid-pregnancy, they're looking for clothes, bedding, shower. And at the end, they're asking questions like into labor, water broke, my newborn. And so this is, I think, really a proof of concept that we can use data from search engines to inform us about medical events. So we did a retrospective study using existing search data, and we identified cases and controls. Now, the ALS cases we defined as basically people who told their search engine, I have ALS. They had to type into their search engine, I've been diagnosed with ALS, or I have been diagnosed with ALS, I had to come up with this, but it's basically this statement. Now, someone might type that in and not have ALS. I understand that. So this is not a ground truth, but it gets us, I think, at least a proof of concept. And in fact, if there are lots of people telling their search engine, I have ALS, when they don't, that will bias us toward a null hypothesis and we wouldn't find anything. We then identified controls who had similar queries overall to the cases, but did not tell their search engine that they had ALS. And we had 50 controls for every case. And then we used AI to differentiate the cases from controls based on their search data. Now, for privacy reasons, we could not look at the content of their search engines, except for what we call ALS-related queries. So we came up with a list of queries that people with ALS might be looking for. For example, Riluzole, Edaravone, fasciculations, and we had a long list of these. And then we had to look at search behavior. So things like how long were sessions and how often was autocorrect used. We identified 285 ALS users. Those users had, on average, over the period we looked at, 9 queries per day. And they averaged 22 ALS-related queries over that time. I think it was a 2-week period, may have been a 4-week period. And we had 3,276 control users. They averaged many more queries, 48 queries per day. And on average, they only looked for 0.6 ALS-related queries. But interestingly, There were about 400 of them that looked for an average of 4 of those ALS queries, and the rest looked for none. So there was sort of a dichotomous population within the controls. So this shows some of the ways that people with ALS differ from controls in their search behavior. People with ALS had used autocorrect more, and the ALS group also used more rare queries, which is interesting. We don't know exactly what the content of this is, just that they're rarer queries. Our power to distinguish ALS from controls was actually really quite robust. The AUC was in the range of 0.9. And the 4 most critical variables were session duration, autocorrect, query likelihood, and whether that query was used by fewer than 5 users in the data set, so ultra rare queries. There are 3 lines on this ROC, and the reason is that we looked at 3 different epochs. So we looked at sort of 2 weeks or a month, and then a year later, 2 weeks or a month, and a year after that, 2 weeks or a month, thinking that it may be easier and easier to distinguish people with ALS from controls as the disease progressed. But it turns out actually it was probably equally simple at any of these times, which is interesting. It sort of begs the question, would this be an early diagnostic? I want to be careful about that. Would this be an early way to potentially identify people with ALS? It is really not about coming up with a diagnostic tool, and I don't want to go to sort of dystopian places with this. The real point here is, can we use data sets that we don't traditionally use in medicine to explore health? Now, to follow up on a retrospective study, because of some of the weaknesses that I talked about, we did a small prospective cohort and to replicate those findings. So we developed a website where people could consent, fill out a brief survey about ALS, and then link that survey to their search results, their search history, without us ever knowing who the person is. So we could do this anonymously. There were 122 people went to the website, 85 consented, 60 completed the questionnaire. Of those, 45 reported that they had ALS. And this is a Microsoft project, so Microsoft research project, so we were using Bing. So only 16 of them had Bing data. So we had a very small group of cases. And then we matched them to 3,750 control users. And we used the model. We didn't build a new model. We didn't tweak the model. We used exactly the model that we had built for the retrospective case control study and applied that here. And we still got a very good AUC. And I think we could have run this longer. It was really became a matter of resources. But even in a small prospective study, we could still separate people from ALS and controls. Again, I think the question becomes, could data like this reduce diagnostic delay for ALS? Is there some way that that could happen? I don't want to, again, it's not so much a use case that I'm saying we ought to be diagnosing people like this, but rather there is a lot of data out there that we're not using for health that we could be. And I'll just say that, we've had meetings with grocery stores because they collect a lot of data about us, and people with ALS change their eating habits over time. There's some complications to using that data, but there are these large data sets that we just don't think about that may have some use in health. In fact, diabetes actually, some diabetes programs are using grocery store data to understand that disease. But if we really want to get at understanding the early parts of this disease, we need to be creative. And so that's one idea, but I think a more concrete idea is a study that we call Prevent ALS. And Mark Array is here. He's really, this was started by Katie Nicholson. Mark Array is really taking this into his hands and driving it forward. The idea is to identify the earliest biological and clinical changes due to ALS and pre-symptomatic gene carriers. So we have a population of people that really will develop disease, or has a high likelihood of developing disease. And we can sort of follow them before they have disease and see what happens first. So the design of this Prevent ALS study, and very briefly, is that we identify an at-risk person, that is somebody who comes from a family with known genetic ALS. We consent them. They can choose to be in what we call the disclosure group or the non-disclosure group. The disclosure group means that they want to know their own gene status, and non-disclosure means they do not. There's a lot of talking that goes into understanding this for the participant. We take that very seriously. We do a screening exam in history to make sure that they don't have ALS and that they are pre-symptomatic truly. And then we test for the ALS causative disease genes, and we return those results. Very, very difficult visits, or at least highly emotional, or we don't return the results if they're in the non-disclosure group. And then we collect outcome measures every 6 to 12 months. And those include blood and spinal fluid for biomarkers, quantitative strength testing, ALS-FRSR, quantitative motor speech analysis, sometimes an EMG, and many other things. We do these in clinic, and we do it every 6 to 12 months. Now, Prevent ALS is actually a combination of two studies. The DIAL study that we've been doing for many years with MGH and WashU, where we have over 200 participants and almost 10 converters, that is, people who have gone from pre-symptomatic to symptomatic. And the family study, which is being run at Columbia University, very similar design, and we're aligning those studies. They have 148 participants. So by joining forces, we automatically essentially double our power in this study, and we'd like to expand from here. But there are some challenges for Prevent ALS. There's a low incidence of genetic ALS, about 15% of all ALS, and people are not clustered in one location, but spread throughout the country. There's no readily identifiable prodrome as there is in Parkinson's with REM sleep behavior disorder. The gene mutations are not associated with a specific age of onset in most cases, as we see in Huntington's disease. And there's a lack of specific biomarkers for early disease, which is why we're doing the study. So we don't really know when to focus our monitoring. So we just sort of see people at regular intervals, but it has to be relatively infrequent. People are healthy, they're living their lives. They may be coming from far away. That's a challenge because onset of clinical weakness is easy to miss if we're doing annual visits. It's very unlikely it's going to happen that day. So there is a real role for digital biomarkers in this study, I think, because we can do frequent, easy monitoring that would help us identify conversion in real time, and then we could bring people in real time to assess their symptoms. And so I think that's a real opportunity for digital biomarkers in this study. So I'll conclude by just saying that digital data is giving us new ways to deliver existing and novel PROs and generating new types of quantitative data and new insights into ALS progression. Active data requires participant effort, but is focused, and analysis can be straightforward. Passive data can yield these very large data sets, but requires really intensive processing. The benefit is it can be analyzed in numerous ways to get novel insights into functioning. I have one more slide after this, and I'll talk about that in just a moment. Quantitative motor speech analysis is actually quite mature, and it's providing quantitative measures of bulbar function. We're already putting it into clinical trials. Cough analysis is newer, but really might quantitate bulbar disease progression. Digital endpoints themselves can, I think, improve ALS trial design, and we've begun to put them into trials. And attention to the usability, the burden, and compliance is critical to success. Now, I've sort of talked a lot about compliance for people in the studies, participants, but I think we have to think a lot about coordinators because they have busy jobs running a trial, as well as project managers who are running multi-center studies. If we don't get all of those parties on board, then these are destined to fail in our trials. So I think we need to really take a deep look at what do each of those parties need in order to make this successful. And then lastly, there are huge troves of data that are completely unmined for ALS and neurology research. And I would just say, let's think creatively about it. So our future directions include sharing existing data to find powerful new analyses. So Anupam Gupta, close colleague in some of these studies, showed us an analysis of accelerometry data, not actually from our studies, but from a group called ALS TDI the other day, looking at submovements, which was remarkable and I think incredibly exciting. Martin S., we'll call him, is a postdoc at JP Onella's lab at the Harvard School of Public Health and is doing an analysis of arm movements using our accelerometry data from Actigraph that is remarkable and paints a very incredible picture of how things change over time for arm movements, not so much step counts. We want to study new and emerging best-in-class devices. So we're starting a new project with colleagues in Belgium and a company called SysNav that makes a wearable device. They actually made the guts for guided missiles and then shrunk those down so that we can use them as a medical device. They actually have their first EMA, the first EMA approval for a secondary endpoint in Duchenne muscular dystrophy, looking at stride velocity. And we want to create ways to incorporate these digital endpoints into trials seamlessly, which I just talked about. But on the platform trial, we have a biomarkers and outcomes working group where we can kind of vet some of these, some of which are not ready to go into the trial. But then the good ones we can bring to the design team and say, this is really important. We should put this in. And that's how, that's how I think we're going to make progress. And we want to incorporate and adapt our digital approach into the PREVENT study. And the one thing I didn't really say is that an approach to healthy participants may be slightly different than an approach to people with ALS. There may be different tolerances for how frequently we ask them questions, kinds of things we ask them to do. And so we need to really be careful about how we do. This work is done in collaboration with lots of wonderful colleagues. So JP Onella, I've mentioned a number of times, is at the Onella lab at the Harvard School of Public Health. He built BIWI. He is a partner in analyzing that. It's just been a lot of fun to work with him, and he's had wonderful postdocs. who have worked on these projects. Jordan Green and Kate Conahan have been longtime collaborators on these projects and many others. And I just value that collaboration very much. It's been, I think, over a decade I've been working with Jordan Green. My colleagues at the MGH Healey Center, Merritt as a mentor to me, but Sabrina, Suma, Doreen, Mark, and Stephen Johnson, who was a fellow here last year. This is one of the most fulfilling parts of my job, is working with these people. Anne-Marie Wills she and I have worked on a few projects together using tech. She is really inspiring because she just doesn't sort of take no, and she's just unafraid to try something new. She's using many of these measures in movement disorders, and it's just been fun to have somebody to kind of compare notes with along the way. We have incredible operations team, data management systems, project management, who allow us to do studies like the PREVENT study and some of these other multi-center studies that I've talked about. I want to mention our industry partners for some of these: MT Pharma Holdings of America, Biogen, and Q Medical. Everything ALS is a nonprofit, which has really taken on doing some of these digital studies in a big way. And I'm on their scientific board of advisors, which is in my disclosures, but I think they're really doing an interesting job of taking some of these early findings and thinking about the regulatory pathway. Ernest Frankel and Divya Ramamurthy have been wonderful collaborators. We have coordinators, many of whom are here, who have worked on these projects diligently, and that's the only reason that we've gotten there, and nurses and nurse practitioners at the Healey Center. And I want to take a special moment to talk about Katie Burke, who is a physical therapist, and Katie has been a partner on all of these projects, as well as the many projects that didn't make it to slides because they didn't work or they're not done yet, never deterred by failure. I think our first project was the Microsoft Connect project. We said, well, look, if you can use it for video games, you can use it to understand ALS, and that's not true. It also turns out it's not that great for video games. They've stopped producing it. So, but in all seriousness, Katie is just remarkable. She knows more about digital endpoints in ALS than probably anybody else in the world. And I want to thank her deeply for all of her help and camaraderie through all of this. Thank you very much. Good question.
Congratulations on this body of work and your leadership in this area. Really global. I'm just kind of curious, where do you see this eventually coming into here, or like what's kind of the path?
Yeah. So Merritt asked the question: do I see this coming into clinical care? And I think that's a great question. And I sort of said at the outset, I'm not going to focus on this so much. I do think that there's a role for this in clinical care. And actually, motor speech analysis is probably the most advanced of all the things that I talked about. We are working with Stacy Sullivan in our clinic now to begin making recordings in clinic and doing some of these very simple analyses that are really at the level of clinical care. So words per minute, we can track that quantitatively over time in clinical care. And it may actually tell us something about when to get people involved with augmentative communication. It may also correlate with bulbar function in a way that we can say, oh, at this point, we need to get involved and have a swallow study on somebody, when words per minute fall below a certain threshold. So that's an example, but I think that there are lots of ways that we can do this. Katie is also working actually on a very simple protocol. It remains research at this point, but to use some accelerometers in the clinic, basically, to see whether that will inform our physical therapy care at all. So I really do think that there's a way. I'd like to think that there's a way to use them for diagnostics. So someone comes in for your first visit, you give them some device, they go home with it, and a month later you have, really meaningful data about how they're moving that might tell you about diagnosis.
I do want to acknowledge that we have almost 80 people connected online in addition to a full room here, which is wonderful. And we have a few questions online. So real quick, can you comment on the challenges in cognitive assessment via digital?
Yes. So I think there are two challenges. And the first one, I just have to bring a little humility, which is to say I'm not a cognitive neuroscientist. And that may be the biggest problem. Having said that, in the study we did with Biogen, the digital ALS study, we did work with people who were really cognitive scientists. One of the challenges was that we took paper and pencil tasks and we put them onto a smartphone. So things like trails A and B. And it's not that they didn't work, they did, but what we think is that the space on the phone is much smaller. And so it's actually easier to do. And so we just didn't get as clear a separation as we thought we might based on results from paper and pencil tests. And then the last thing is that there are cognitive changes in ALS. They tend to be associated with frontotemporal cognitive change. It's only dementia about 5% of people. It's subclinical in a larger percentage of people. But how we get at those subtle changes is a bit of a challenge.
I know we're almost out of time, but we have 2 more questions. For digital biomarkers that aren't obviously functionally important, like speech pauses, how do you make the argument to use them as outcomes? Is it enough to show correlation with traditional measures?
Yeah, that's a great question. So we have showed correlation with traditional measures like ALS-FRSR, but remember, ALS-FRSR has one question about speech, and so it's not a very robust comparison. And that's where Kate Conahan's idea of using this more robust PRO about communicative participation, I think, comes in. So we can say, okay, the percent pause time, the speaking rate correlates with an inability to participate in life using verbal communication, or a challenge with using verbal communication. I think that's how we begin to make that case.
Last online question. To follow on Merit's question, can you identify subsets of patients' unique phenotypes that might inform a personalized medicine view of ALS?
Yeah, this is a really interesting question. I think we've said that the disease can begin in the bulbar region, and the cervical region, the lumbar region, respiratory, very occasionally generalized too. And so I think when I think about how we might use digital measures to look at subtypes, I think about these clinical subtypes, people who have just bulbar symptoms or just cervical, arm symptoms or just leg symptoms, and how those symptoms are spreading. And I think actually if we could quantify that, it would tell us something even new about the disease. So, we have a hard time predicting how the disease will progress, but maybe there are tells in more quantified digital data that says if something is progressing this way in one region, but not spreading to another region, that's a good sign they'll have slow progression. Or if the disease is spreading even subtly from one region to another, we can predict maybe rapid progression. So I think we could subset people and also learn something from that kind of analysis.
That was all for online questions. I don't know. I think now we are out of, we have one more question.
So James, if you put your Elon Musk hat on for a second and thought about how you could deploy this more widely, even asking Alexa or Siri, probably a lot of things are already analyzed. That amount of text that you can apply to the whole population and say, who is an early, ALS, someone on that trajectory, or for Alzheimer's, who needs antibodies for him. It seems like this would be a great framing sort of study.
I love this. So I love this question. The question is, can we first put on my Elon Musk hat? Let's put on the hat more the suitcase of money. I like that. But put on the Elon Musk hat, think big. Could this go to sort of a population-based kind of analysis for ALS, but also maybe Alzheimer's or common diseases? And what about using something like Alexa? And actually, I have had some meetings with folks at Amazon over the years, as well as people at Apple. I think there are some privacy concerns. It's very interesting that we are, we're very happy to use our data. And this is actually true. I would say this is true in my experience as well. People are happy to use their data to sort of get targeted ads. They're more uncomfortable about a physician getting that data. I think in part, I've thought a lot about this because it sort of doesn't make a lot of logical sense, but it makes a little bit of emotional sense. And I think part of that is that when you think, okay, there's going to be a physician looking at this data, you can picture that person looking at your data and somehow learning something about you. Whereas, if it's going into the cloud and somebody's selling you, green pants because of whatever you searched, that seems sort of fine and amorphous. The reality is when we talk about analyzing big data sets, nobody's sitting down and looking at, what that search was for or where a person spent time. So I think some of that it can be waylaid. But I actually do think that there is going to be a move into this and there will be privacy parameters put around it, and there'll be sort of opt-in programs. And I think these far-field microphones, like what's in Amazon Alexa or Hello Google or whatever, really will have a use. Yeah.
Thank you, James. Thank you.